We left off on Friday. We went through the different types of formed elements. We went through erythrocytes, and we're going to continue to go through leukocytes and platelets today before we get into hemostasis. Okay, so we went through hemopoiesis, the generation of blood cells. We went through the different types of erythropoiesis and leukopoiesis, and now let's talk about leukocytes. These are your white blood cells. All right, so your WBCs. They are the true cells of the formed elements. The other two, red blood cells and platelets, are not necessarily true cells. Okay, they are red blood cells don't have a nucleus. They don't have organelles. Platelets are cell fragments. Therefore, they are not true cells. Okay. Leukocytes, or collectively, they work to fight against infections and pathogens. All right, so they'll recognize and help to counteract potentially disease-causing agents, bacteria, viruses, cells that are broken down, dead cells, cancerous cells, or cells that are growing uncontrollably, you name it. Okay? To do this, they are kind of what we call sentinels. They travel throughout the bloodstream, although most of them are not found in the bloodstream. They are actually found in other places, the bone marrow, the thymus, lymph nodes. That's where the bulk of the white blood cells are found, but they do travel throughout the bloodstream as necessary. All right? With that, they are what we call modal and flexible. Okay? Most are found in tissues, not blood, especially the lymph nodes and the lymphatic tissue. And we'll talk about the lymphatic and immune system in a couple weeks. Okay? But they can exit the vessel wall by a process called diapedesis. All right? And that diapedesis is where they squeeze through. So it ends up looking something like this. This is your vessel wall that's coated by endothelium. All right, so the red cells are the endothelium. Here's the vessel wall, and you have this endothelium here. And a white blood cell, actually, as it goes around, a lot of times it is rolling along the path or along the side of the vessel. Okay? So it's rolling along the side of this vessel. Eventually, you end up at a point where here's something that's happening in this tissue. And this white blood cell, or this bacteria that's down here. Here, I'll put it up here so you can actually see it. Here's a little bacterium, okay? And this white blood cell rolls through, and eventually it's going to, maybe there's a chemical that's released by this bacteria that attracts this white blood cell to start to squeeze through and enter the tissue around it. Okay? And that process of squeezing through the edge of the vessel wall is called diapedesis. Okay? Because they are this amoeboid and flexible-like structure, they squeeze through the edges of the endothelium that are lining the vessel. They exit the vessel wall, and then they go into the tissue, eventually coming around here, and engulfing, right, becoming a macrophage, engulfing the bacterium, right, and digesting it inside a lysosome, and then removing the pathogen from the tissue. Okay? The process of attraction to that area by some chemical is called chemotaxis. All right? So chemotaxis can be both positive or negative. In the case of white blood cells, this is positive chemotaxis. Something is going to be released either by the pathogen, by dead cells, the presence of dead cells or cell fragments, chemicals within the cell. Something's released into the environment that signals the white blood cells to come here. Right? It's like pulling the fire alarm. something's wrong, come here, right? So it comes, and then eventually it attracts it to it and brings the white blood cells to the area. So you get a cut, you get a small infection, okay? Sometimes that cut doesn't heal right. It gets kind of what does it look like? What's the cut? I don't know. My daughter plays softball, so she slides in the second base and gets a small cut, and she gets some dirt inside cut. And eventually, what does it look like?
It gets red.
Gets red, gets swollen, it becomes inflamed, okay? And then sometimes it ends up with some gunk, right? Some like pus that's there, okay? So that red, swollen inflammation, that is there for a reason, okay? The capillaries become leaky, okay? They open up so it allows for the white blood cells to leave the vessels easier. You get swelling, that's more fluid that's going through there. The white blood cells go there and clear out any cellular debris that's been present, okay? And so all of the broken blood cells or broken cells of the skin that's there, they also release chemicals that will attract those white blood cells there. And what you end up with is this kind of pussy stuff. That is usually dead white blood cells or cells that have been removing any pathogens and eventually it's left over. Okay? You don't want it to look green because then that's a problem. If it's white and kind of yellowy, that's okay. That's pretty normal. All right? So let's go through the different types of white blood cells. You did this in lab pretty extensively if you went through and identified which ones they are through Histogram Plus or whatever the software is called. Basically, we have two different categories. You have granulocytes and agranulocytes, and that is presence. That determine whether or not there's visible presence of cytoplasmic granules. Okay? And so we have neutrophils, basophils, and eosinophils. Those are positive for granules. Okay? So those are granulocytes, and lymphocytes, and monocytes. You cannot see. All right? They have cytoplasmic granules, but they are not visible by light microscope. All right? So we call them, we separate them, categorize them as agranulocytes. Okay? Neutrophils are the most abundant type of white blood cells in a normal blood sample. Okay? When you have an abnormal blood sample, you could have different numbers. Okay, so this is what it looks like. I will go through this real quick. So you've all seen pictures of each of these. Neutrophils have a 3 to 5 lobe nucleus. You may see a very light purple staining of the cytoplasmic granules. Okay? But with a lobe nucleus, that's pretty indicative of a neutrophil. Okay? Eosinophils have a bilobe nucleus, 2 parts, and their cytoplasmic granules are stained a darker red. Okay? This is named, eosinophils have the name eosinophil because eosin is the dye that is used to stain them. Okay? Eosin, phil, what is philia suffix of? What does that mean? Attraction to or likes to do something, right? If something's hydrophilic, it likes water, right? So eosinophil is something that likes eosin. That's why it stains red, okay? And then you have basophils, which are the nucleus is like S-shaped and has a very dark purple cytoplasmic granules, right? Basophil, it likes the basic stain, right? The pH 7.4 plus. That's what the stain is, and that's for a different purple color. Okay? Questions on those? We're going to go through what each of them do. The other two, the agranulocytes, are lymphocytes and monocytes. Okay? Lymphocytes, mainly a spherical nucleus, takes up most of the volume of the cell with a very small edge of cytoplasm. And then the monocyte has a U-shaped or a slightly indented part of the nucleus, right? So it's not necessarily spherical. and there's more cytoplasm that takes up less of the room of the cell. Right? The monocytes are usually larger. Monocytes are the ones that will exit the bloodstream and become macrophages in tissue. Okay? And each of these different types of white blood cells is responsive to different conditions. Okay? So, go up here, and you can look at the different parts. It gives you an idea of what they look like, their basic function. And you'll see that there's a couple different ones that are important. Neutrophils are a prototypical white blood cell. Okay? They phagocytize pathogens, especially the bacteria. So the example I showed was probably a neutrophil, right? And they also release enzymes that target pathogens. Eosinophils actually will phagocytose or phagocytize antigen-antibody complexes. Antigen-antibody complexes. So give me an example of when an eosinophil will be utilized from something we talked about last week on Friday. Where's an antigen-antibody complex going to come into place?
Like if you have like B blood and you get different blood.
All right, so if you're B-type blood, you have A-type antigens. If you get A-type antibodies and you get an A-type blood from someone else, right? You don't get a non-compatible blood type. You will end up with an antigen-antibody reaction, and eosinophils will react to that. Okay, and there's also allergens. Eosinophils are involved in allergens, as does basophils. They release histamine, a vasodilator, and there's some inflammatory reactions that are involved. Okay, and then lymphocytes. Lymphocytes make antibodies. Right, they will be induced to become different types of cells called plasma cells that produce antibodies. All right, and then monocytes. They are. Why is this still on there? It'll go away in a second. Monocytes, they become macrophages, right? And they phagocytize pathogens, bacteria, viruses, cellular fragments, etc. Okay? And they all have different percentages. You'll see neutrophils are the most abundant, right? Then lymphocytes, then monocytes, then eosinophils and basophils, which are less than 1% of the whole total amount of leukocytes. Okay? In lab, you did a white blood cell differential count. You counted the white blood cells through that histogram plus. You went through 100 cells, right? And you identified, you differentiated them from one another. You didn't just say, oh, that's a white blood cell, tick, 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 slash, whatever. You actually said it's a neutrophil, it's a monocyte, it's a lymphocyte, it's eosinophil, it's a basophil. You differentiated. That's a differential count. Okay? And so when you come up with that, you come up with a normal amount of, a normal range of where neutrophils should be, or where monocytes should be, where lymphocytes should be, right? And a clinician can tell that if you have elevated or lowered beyond that normal range, something's going on. It's indicative of some type of problem systemically. Okay? So if you have elevated neutrophils or an elevated white blood cell count, probably a good chance you have some type of infection, cold, virus, flu, whatever. Okay? As an example. So we have different characteristics. We could talk about that white blood cell count. We can have leukopenia. Leukopenia is a reduced number of white blood cells, total white blood cells. Okay? And what does that do? Well, if you don't have enough white blood cells, your immune system is compromised, and you have a higher risk of infection. Okay? This might be someone who has leukopenia, might be taking immunosuppressants for maybe they have a kidney transplant or something like that. Okay? Leukocytosis. That's an elevated number of white blood cells. All right, recent infection, stress can also induce white blood cell count. All right, which can be good or bad. All right, good temporarily, like within the short term. You have a little bit of stress, you have a boost in your immune system in case something else, but over long term that could cause problems. All right, and we have neutrophilia, an increase in neutrophils above normal. All right, this is associated with bacterial infections, stress, and tissue necrosis. What's necrosis? Death, right? So the tissue, some type of tissue damage is ongoing. All right, and then you have neutropenia. So philia is above normal, penia is below normal. Okay, and this can be attributed with drug, radiation therapies, right? So you have chemotherapy, I have an anemia for red blood cells, and a neutropenia, a diminished immune system. And then lymphocytosis, an increase in lymphocytes. And this is caused by viral infections, all right, or chronic bacterial infections such as chronic bronchitis, okay? And then eosinophils numbers, they rise during allergic reactions, parasitic infections, and some autoimmune diseases. Why didn't I include basophils in here? What's the normal percentage of basophils? Like less than 1%, right? So if they go up, they're going to go from 1% to 2%. It's not going to be a big deal, okay? And they're also involved in some allergic reactions as well. Okay? So hopefully what you get from this is I can give you a case study of saying, all right, well, let's go through this. This morning, I have the fun morning of getting my daughter ready for school. The older one off went with the wife, right? She wakes up earlier, she goes to middle school, but I get to wake up and take care of the first grader. Yay. Okay? She didn't wake up the time she normally does. She was kind of felt nice and cozy in bed, so I had to actually go upstairs and wake her up. So 7:45 this morning, I'm like, come on out of bed, whatever. And she comes downstairs and she says, I don't feel good. My stomach hurts. Oh, no. Okay, maybe you're just hungry. So get some food in her. She felt better, brushed teeth, ready to get dressed. All right, we're out the door. And she's like, I don't want to go to school. I don't feel good. I was like, well, how about this? Maybe it's just breakfast. Go to school, and if you feel bad, you go to the nurse, and the nurse will call mom, and mom will come get you, or your grandmother will come get you. Okay. Off I went, driving to work. Here we go. 20 minutes in, get my cell phone rings. This is Eggermont School. Violet doesn't feel good. She's got a fever of 99.1 and her throat hurts. Okay, I'll be right back. I'll go. So I went back and picked her up and then brought her. What do you think might be happening? Some type of infection. Okay, fever is one of the hallmark signs of infection. Okay, now her throat hurt. What's the common bacterial infection that's ongoing now? Let's common bacterial infection. The flu is not bacteria. Strep throat. Okay, now a bunch of her classmates had it last week, so I'm hoping that that's the case and it's not the flu. Okay? So if it is strep throat, what type of leukocyte would you expect to be elevated? Neutrophils. Why? Because it's a bacterial infection. Okay? If it is the flu, what type of leukocyte would you expect to have? More lymphocytes. Okay? And how long does the flu usually take? It takes a couple days, right, for you to kind of go through something, because your lymphocytes have to be exposed to the virus, and then it takes a couple days to kind of increase antibody production. All right, good. So, the last formed element that we need to talk about are platelets. And platelets are what? Technically, what are platelets?
Cell fragments.
They are cell fragments. Okay? They're also called, we call thrombocytes. All right, and they're involved in what? Clotting. Okay? Really, they're involved in the process of what we call hemostasis. All right? We're going to talk about hemostasis today. What was that? Drag and drop, touch and hold. Okay. So hemostasis is the stoppage of bleeding. Guess what? We're not perfect. We're not going to go through life and never going to have some type of bleeding. Okay? So, luckily, we've also adapted a process to stop bleeding. Okay? And the process of hemostasis is really three overlapping, I don't want to say procedures, or let's say phases. They call them phases. There's three overlapping things that occur. Okay? Vascular spasm, platelet plug formation, and the coagulation phase, or clotting, or coagulation. The blood coagulates. We're going to go through how this happens. Okay? In general, they're kind of, the three phases are actually diagrammed here. So first, there's some type of injury. A vessel breaks. Okay? The vessel breaks, blood starts to come out. When the vessel breaks, it also exposes fibers that make up that vessel. There's pieces of cells that have been broken. There's collagen fibers that are exposed. And with that, that starts the hemostasis procedure. Okay? So the first part is a vascular spasm. Vascular means blood vessel. Well, the blood vessel itself, and we'll go through this when we go through the vessels in 2 weeks, okay, is actually a cylinder, okay? And inside the cylinder, there are two layers of smooth muscle that make up that cylinder, okay? One layer runs longitudinally, okay? Then the other layer runs circular, okay, around. So what ends up happening, you break the vessel, those muscles, those smooth muscle cells, they contract. Okay? They contract. And what that does, it prevents, or helps to prevent, any excessive blood loss. Think about it like this. Yeah, you all know what a garden hose looks like? Good. Okay? You know, at the end, the sprinkler. Okay. What happens if I take a knife and I cut the garden hose? And I'm trying to cut it off, like I cut it and I open it. Now the water's going to be pouring out all over the place. If the water's on, what's one way of stopping water coming out of the hose? I could kink the hose, right? And do it that way. That's what's happening here. The vessels are trying to prevent liquid from flowing through the opening and losing more volume. Okay? Vascular spasm. that's pretty straightforward, right? We're trying to restrict flow so that we don't have excessive blood loss. However, next, we have to repair the injury, okay? We have to put a duct tape around the cut in the garden hose, okay? So first, this is the next step, is what we call platelet plug formation. The vessel now is broken. So here's our vessel that has been broken. And there are the ends of the vessel, there are these fibers that are exposed. Collagen fibers are exposed. And we have red blood cells that are still flowing through here, okay? And then with that are also what? Red blood cells. What else are in the blood? White blood cells and platelets, okay? So the platelet plug formation is a positive feedback mechanism, okay? That once started, it enhances the stimulus and makes more of the event to occur, okay? So normally when uninjured, the platelets aren't going to be activated, they're just going to flow through. But now we have, here's a platelet is going to bind to our open vessels, okay? So the collagen fibers in the vessel wall are exposed, a platelet binds, okay? Platelets start to stick to these fibers. So once one sticks, that actually causes more of them to stick. So now I'm getting lots of these, and now more of them are stuck, and now more of them stick more. Eventually what ends up happening? The platelets aggregate and they close off the injury. Okay? That kind of stops blood from being able to flow freely out. Is it completely clotted yet? No, it's not. Okay? We have not repaired the vessel. We just stopped the flow from coming out. So the good analogy is there's a cut in the garden hose, water's on, I've kinked the hose, I need to keep it going, so I take some duct tape and I wrap it around. Now I've stopped any other water from coming out, okay? Or any other fluid from, I'm reducing the amount of material. What do I still need to do? Is the hose or the vessel still, can that hold fluid to go through it yet? Not really, okay? Not completely. Is the vessel repaired completely? No. Now I have to do something to start the repair process. So platelet plug formation is a positive feedback mechanism with the help of something called von Willebrand factor. You'll see that in a minute. Okay, so why is it just happening right here and not all the way along the vessel? Well, the normal cells that are the healthy cells that are here in this part of the vessel, they're producing stuff that will inhibit platelet plug formation. So it only happens where the vessel's broken. Okay, it only has to happen where it's necessary. Okay, aggregates and closes off the injury. There's a whole different set of process and steps of the process. Now, you don't need to know what serotonin and thromboxanes that are doing. I don't care. Okay, understand that this is a process. It's a stepwise process. All right, and there are enzymes involved. Right, and there's procoagulants that stimulate coagulation. And eventually what has to happen, this stimulates mitosis, which then begins the repair of the blood vessels. Okay? So a couple things that are still happening. We're still going to prolong vascular spasms, prevent excess blood flow, platelet plug formation, and then it starts the next process of coagulation. In some cases, you end up with, some people suffer from something called thrombocytopenia, or low platelet count, which impairs all phases of hemostasis. Okay? So that underlines the importance that platelets are a link between vascular spasms to coagulation. You need the platelets. If you don't have the platelets, then the coagulation isn't necessarily going to happen, or it's going to be impaired quite a bit. Okay? This forms pretty quickly, within a minute, but it's prevented from getting too large by prostacyclines. So why doesn't it get too large? So thrombocytopenia. So that's the platelet plug formation. We go to the third phase, coagulation phase. All right, and this is more involved and involves producing a network of fibrin. Okay? Fibrin. Now, when we started blood, we said blood was what type of tissue? Connective tissue. It's a fluid or liquid connective tissue. What is connective tissue? Is characterized by, all connective tissue has a couple things, a couple characteristics that they share. What are they? Cells suspended in a matrix. So we have cells, white blood cells, red blood cells, suspended in a fluid matrix of plasma. Okay, what else? This is a ground substance that's composed of what? There's ground substance. They have fibers. Right? And up till now, blood hasn't shown where we have fibers. Okay? So this is the part where fibers are involved in the coagulation part. Okay? So fibrin is an insoluble protein, but it exists in blood plasma as fibrinogen. Well, now we'll do it in black. Fibrinogen. No, fibrin, that's an I, O-gen. Okay? This ending, N-O-G-E-N, nogen, or ogen, it means an inactive form of something. Okay? So fibrinogen is an inactive form of fibrin, which is soluble, it's also the soluble form, in blood plasma. Okay? So as blood is flowing through your veins, the capacity to clot and coagulate is there. It's always there. Okay? It just needs to be activated through this process. Okay? The mesh that's going to be formed by this is going to trap the erythrocytes, leukocytes, platelets, and really form a clot, a plug, right? A permanent plug that's going to allow for the vessel walls to grow back and repair themselves, and then it'll be broken down and then replaced by healthy vessel walls. This is a scanning electron micrograph of what a clot looks like. You can see the fibers. There's some platelets attached. The erythrocytes are stopped. Right. This is kind of what a scab looks like after it stops bleeding, right, and then starts to heal a little bit. There are lots of substances involved in this coagulation phase. All right. You need calcium. Calcium is necessary for this. Vitamin K is necessary. There's a bunch of clotting factors. Well, you'll see on the next, there's like 14 clotting factors, right? Some of them they're all important, okay? Factor 1 is fibrinogen. Their factors are named based upon when they were discovered, or when they were realized to be part of the clotting cascade, right? Fibrinogen, prothrombin, we're going to talk about them in a bit, okay? And some of them are proteases that cleave other factors, right? So if fibrinogen is floating around in blood plasma as an inactive form, we have to find something that is an enzyme that's going to convert it from an inactive form to an active form, from fibrinogen to fibrin. That enzyme is a protease. It's going to work on a protein and do something, okay? And there's a whole cascade of events that occur, right? Lots of things. And vitamin K is also involved in this. Here's the factors. This is in your textbook, table 18.8. Put it in here just kind of so you can see there are 13 factors listed there. Some important ones: calcium, right? Prothrombin, tissue factor, okay? All of them are involved. There are, and then to the far right-hand corner, you'll see that there's a clinical syndrome if the clotting factor is not there. If these clotting factors aren't there, what doesn't work? What's impaired? The
clot formation?
The clot formation, the coagulation phase is impaired. So then what ends up happening to that person? They have excessive bleeding. Okay? And if you go through, there's some that aren't known, but then some other ones that are important. Hemophilia, we'll talk about A, B, and C. There's afibrinogemia, right? Which are usually, these are all usually genetic disorders. Okay? So let's talk about the initiation of the coagulation cascade. How do we start this? And how does it get to the part where we're converting fibrinogen to fibrin? Because that's where we want to go. Okay? So there's actually two different pathways, what we call intrinsic pathway or an extrinsic pathway. What's the difference? One's either initiated on the inside of the cell, inside of the vessel, or from outside the vessel. Okay? The intrinsic pathway. You see phase one to the left over here, right? This is your intrinsic pathway. Here's your extrinsic pathway. Intrinsic. The vessel endothelium ruptures, exposing the tissues, the collagen, like we just saw. Okay? So you have platelets, plug formation. They provide the sites for the factors. Then the factors start to go, right? You have all these arrows are just showing activation. So one factor activates the next, which activates the next factor, and there's a stepwise progression. Okay? Ultimately, it comes down to here, which you end up with this part. This is the big part right here, the important. Activation of prothrombin activator. Okay? The extrinsic pathway, there's a tissue cell trauma, exposes blood to tissue factor. Okay, so tissue cell trauma. What's happened? The cells of the vessel have been broken, releasing tissue factor to the blood. That now starts this cascade. So really, we talk about intrinsic and extrinsic pathways. They're really not happening exclusively of one another. 99.9% of the time, both of these pathways are happening simultaneously. Okay? One's a little faster because of the numbers of steps that have to occur. Right? You look to the left here on the left side, there's a number of steps that have to occur in order for this to happen, eventually getting down to prothrombin activator. There's less numbers of steps that have to happen over here. Okay? So this one's probably a little bit faster. Tissue cells are broken, and you end up immediately tissue factors released, and then that cascade starts. Bottom line is they both, intrinsic and extrinsic pathways, converge and produce prothrombin activator. Okay? Now I said that fibrinogen was an inactive form of an enzyme. Okay? Well, guess what? Prothrombin is also an inactive form of an enzyme. Okay? What do you think prothrombin activator is going to do? It's going to activate prothrombin. Okay? We're going to see that in the next part. So you end up, the intrinsic and the extrinsic pathways, they have different steps, but they converge on the common pathway, right? And it comes down to a common, a shared protein at the end, prothrombin activator. Okay, so we end up with this, right? We end up down here, prothrombin activator, okay? That activates prothrombin to thrombin. And then thrombin converts fibrinogen to fibrin. So let's just draw that out. So here's our, oh no, I don't want that. Our, we'll say prothrombin activator. That's a protease. Okay? Here's our prothrombin, which is an inactive form of an enzyme. I said prothrombin activator is going to activate or cleave prothrombin to thrombin. Thrombin is an enzyme that's going to convert fibrinogen to fibrin. Okay? Fibrinogen to fibrin. Oh, can you guys see? I know there's some open spots. All right? But that's the cascade. So we have these two pathways, intrinsic and extrinsic pathway, that are activated upon vessel damage, that converge on prothrombin activator, that starts this. Eventually you get to the point where fibrin is produced. Okay? This, again, is a positive feedback mechanism. The end point is what? There's a clot that's formed. Bleeding has stopped. We've talked numerous times about negative feedback and positive feedback homeostatic mechanisms. Clot formation and childbirth are the two best examples of a positive feedback mechanism. There's a defined endpoint. The clot is produced. The baby is born. Done. There's no more bleeding. There's no more birth or fetus to be pushed out of the birth canal. Okay? Here's what it looks like. Right? There's some calcium is involved in there. So if you were to take calcium out of the picture, calcium's been involved in a couple different parts of this. Okay? Take calcium out of the picture, what happens? Clotting doesn't happen. Clotting doesn't work. Okay? And so the research into this is important because sometimes we don't want clots to form. We'll talk about that in a minute. Okay? This is a picture that's actually in your book. I like the other two better because it takes all the other stuff out. Okay, but you see intrinsic and extrinsic pathways converge into a common pathway at factor 10, also known as before prothrombin activator, and then prothrombin to thrombin. Thrombin converts fibrinogen to fibrin, and you get the polymer and the mesh that's formed. Now that clot stays and you have a scab that's over the vessel, eventually, at some point in time, that needs to go away. Okay? Two things that to get rid of that. There's clot retraction and then fibrinolysis or fibrinolysis, depending on where you put the emphasis on the syllable, whatever. Okay? So the clot is going to retract. Meanwhile, like as soon as the vessel is broken, it actually stimulates the cells around it to start undergoing mitosis to repair what's there. Okay? And that's gonna overlie the clot itself. Okay? So the clot retracts and then it's degraded by an enzyme called plasmin. Usually it happens over a number of days. Okay? And so blood as a tissue balances clot formation with clot elimination over time. Okay? And imbalances in that could lead to excessive bleeding. Okay? And there's a whole different things. Damaged vessels, atherosclerosis. You could have fun things like abnormal clotting that occurs. Okay? So let's go over some quick bleeding and clotting disorders that occur. We mentioned hemophilia. Okay? They are genetic in origin, most in X-linked recessive pattern. We have thrombocytopenia, which is a platelet deficiency. Okay, this could be due to increased breakdown or decreased production of platelets. All right, and it may occur with some bone marrow infection or cancer. There's some viruses that also target that. And certain drugs interfere with clotting, so they cause bleeding. Aspirin, okay? Aspirin, ibuprofen, they cause bleeding because they actually inhibit some of those enzymes that start the cascade. Okay? Some of those enzymes that start the cascade, that thromboxane, those enzymes are inhibited by those drugs, and they can cause bleeding. All right? So for pain after a trauma, they don't give you aspirin or ibuprofen because they'll give you Tylenol. Tylenol is a different type of pain. It targets something else. Okay? Warfarin, ginkgo, heparin. Okay? Heparin is another one. I'll write that one down. Heparin. There's a EDTA. Okay? EDTA is it's used as a preservative in foods. Okay? Now you don't take enough of it to start bleeding all over the place, but what it does is EDTA is what we call a calcium chelator. It binds calcium out of solution. So if it binds calcium, it's going to stop calcium from doing what? Working in this clotting cascade. Okay? So a lot of times when you go get a blood sample drawn, they'll put it in those different colored tubes, green top, purple top, blue top. Well, the purple top have heparin in it and prevents clotting. It prevents coagulation. Because if you took an actual sample out and you just left it on the table, eventually it's going to form this kind of clumpy, gross mess. Okay? It's going to coagulate into a junk of stuff. Okay? But if you want to keep it as fluid blood, you add some anticoagulants to it. And then you can do the centrifugation and spin it down and look at hematocrit and all the other stuff you want to do for clinical use. Okay? In some cases, we have hypercoagulation that occurs, which can lead to something called a thrombus. A thrombus is a blood vessel clot in an otherwise unbroken or normal vessel. Okay? A thrombus. So, one of my favorite baseball players years ago had a blood clot in his elbow, and he had to have a surgery for it. What's the problem with a blood clot in your vessel that's not otherwise broken? What does it do? It stops blood flow. Okay? And the problem with that then is if that clot leaves the part where where it's stuck, where can it go? Well, it can go to a lot of different places. Right? If it's in an arterial blood or venous, if it's in venous blood and it dislodges, where's it going to go? It can go back to the heart and cause a big problem. Or it can go to the lungs and cause what's called a pulmonary embolism. Okay? And so this can cause breathing problems and even death. There could be environmental genetic causes as well, birth control pills, prolonged inactivity. One of the reasons if you're going a long flight, right, they tell you to get up and they tell you to contract your leg muscles or get up and walk around because you're there for a long period of time. The increased pressure of a plane causes blood to start to pool in your legs, which can cause a blood clot, and then it moves around. Now, bleeding can occur. Your sympathetic nervous system responds to that by doing a couple different things. Think about it like this. What's the response? You're going to lose the blood, right? What does your blood want to do? Your body wants to conserve it, right? So what's going to happen? Blood vessels are going to constrict, and blood's going to be diverted to the important organs, lungs, stomach, liver, right? Heart. And it's going to help maintain blood pressure. Your heart rate's going to go up, okay? Your force of contraction is going to go up. So someone who's lost a lot of blood, they have this really high heart, we got tachycardia, and but they might be pale in appearance. They might be cold and kind of clammy to the touch. That's all because their sympathetic nervous system has kicked in to do these things. And then finally, just some stages of blood development. Hematopoietic stem cells are found where? Where are blood cells formed? in the bone marrow, in the red bone marrow, okay? And then over time, your red bone marrow is replaced with fat as you age. So as an infant, the red bone marrow is found throughout long bones, but as you age, the long bones are filled with fat and become yellow bone marrow, and then the red bone marrow is only found in flat bones and in the heads of long bones. Okay? Questions on that? That finishes out chapter 18. I will post your first Connect assignment tonight. Okay? It won't be due for probably like a week and a half. Okay? I'll figure out a due date when I post it. Okay?